Hello and welcome back to another biology episode. Today we're talking about Mendel's laws,
which are the foundation of modern genetics. Gregor Mendel was a monk and he lived in a monastery in the 19th century. He conducted a lot of experiments with over 5,000 pea plants and he generated his nowadays very popular hypothesis. As long as he lived, he died at the end of the 19th century. His rules were largely ignored and they were rediscovered at the beginning of the 20th century. So he died before he knew how important his rules would become for modern genetics. First, we need to clarify some terms that will be essential for understanding these topics. One is allele. An allele is a genetic variant which usually has two forms, so you inherit one from your father and one from your mother. But there can also be more. For example, for blood types there are more. An important concept, it's not really about the words, it's about the concept, is dominant and recessive. So if the dominant allele is present, this means that the phenotype, so how an organism looks like, is defined by the dominant one. Recessive means, as the name suggests, that you can't see it. If an organism is heterozygote, which means that he has two different alleles. So let's talk about the three rules. Actually, if you hear the name, at least the first one is clear. So it's called law of dominance and uniformity. This means if the parent's generation is homozygote for a trait, which means that both alleles are the same, so they're purebred, then the first generation, so the F1 generation will all look alike. So the recessive one, as we've just said, is masked by the dominant one. It's also possible that the features are mixed in the F1 generation, as none is dominant over the other, and this is called incomplete dominance and an intermediate inheritance model, which you can see here, shown by the red and the purple flowers, which together have an offspring with blue, which is a blue flower. So the second law is the segregation of genes. Here we're talking about the F1 generation, which are heterozygote, which means that their alleles are not the same. So in the F1 generation, all of them have one dominant and one recessive allele. So if we're now looking at the F2 generation, we're getting one plant, so one red one, three red ones and one purple one, which means that 1 out of 4 gets two recessive alleles and therefore displays the recessive phenotype. So the ratio of genotypes is 1 to 2 to 1, but for the phenotype it's 3 to 1 for a dominant recessive model and 1 to 2 to 1 for an intermediate model. So, and then of course there's the third law. So far we've just looked at one feature, which in our case was the color of the flowers. Now we're talking about independent assortment, where we take a look at two traits. And so for the F1 generation, again, the uniformity rule applies and therefore they will look alike. But for the F2 generation, the ratio for the phenotype is 9 to 3 to 3 to 1. And I'm pretty sure you will understand it if you take a closer look at the drawing, at the figure. Maybe you can pause the video or even try to draw it yourself to get a better feeling for this. So, and another important point that I want to highlight here is that even though the rules are the basis of modern genetics, and I'm pretty sure you'll learn them in school or hear about them the latest at university, is that in general inheritance is a lot more complicated than that. So these rules do not apply to many traits that can be observed in nature. So just keep that in mind, that it's important to understand these concepts, but in reality it's all a bit more complicated. For example, there's something called linkage disequilibrium, which means that alleles are inherited more often or less frequently than expected, and if they were independent and associated in a completely random way. So often genes are not, or alleles are not associated in random ways. I hope you liked this video and see you in the next one.